Hello, everyone. Thank you for joining us today on this webinar on how to enable next generation IVD devices with nanoMIPs. It is lovely to see so many of you coming into the session now. I can see we've got quite a few people just coming in still. So while everyone joins, I'll just give you a quick navigation of the system, just in case you're not familiar with Zoom webinars. So we'll have a Q&A session at the end of this, at the end of the presentation. If you have any questions throughout the presentation, you'll see at the bottom of your screen there is a Q&A box. Simply click on there and ask your question, and we will address as many of these as we can at the end of the presentation. There'll also be the opportunity to participate in some polls throughout the presentation. These will pop up on your screen automatically, and you can select your answers and press the submit button, and then we will share the results with everyone in the session as well. So I can see we are beginning to fill up here. We've got quite a lot of attendees now signed in. So I will introduce our speakers and then hand over them to get us started. So speaking today, we have Dr. Francesco Canfrotta, who is Head of Chemistry at MIP Discovery. He's joined by Dr. Ashutosh Kumar, who is Head of Technical Operations, and Alan Thompson, who is our Chief Scientific Officer at MIP Discovery. So I'll hand over to you, Francesco.
Thank you, Kelly, and hi, everyone. So today's agenda is going to focus on the current market needs for the next generation devices. We are then going to delve into synthetic affinity reagents with a particular focus, of course, on molecularly imprinted polymers. We're going to discuss how they are made and the advantages brought by them. And then we're gonna bring some examples on how nanoMIPs are employed in sensor devices. At the end, we're gonna have a Q&A session, as mentioned by Kelly, where we're gonna address any questions you may have. Now, starting from the challenges, what are the current challenges of the next generation IVDs? Well, they can be summarized by the following 4 points. First of all, multiplexing. As you can imagine, the end user usually wants to detect multiple target molecules simultaneously. And this is ideally regardless of their molecular size. So it ranges from ions to small organic molecules, peptides, or proteins. The second point is early diagnosis. This is usually achieved by the use of high affinity reagents, which would in turn allow improved sensitivity. Third point is a rapid response to future pandemic situations, as we've witnessed with the current COVID-19 outbreak. And finally, speed of commercialization for sensor technologies. Now, how can synthetics address these challenges? Well, regarding the first point, regarding the multiplexing aspect, synthetics do not require an immune response, and because they are produced in the lab, they can be deployed against virtually any target molecules. Again, ranging from small molecules such as ions or peptides to larger entities, even up to whole viruses. This in turn allows multiplexing to be achievable. Synthetics do avoid the antibody lottery because, once again, they are produced in the lab and we have complete control over the chemistry behind them. We can essentially design them in a way that they achieve superior affinities and selectivity. Furthermore, they can be developed in as little as 8 to 12 weeks, as we've demonstrated with one of our products, the COVID-19 nanoMIPs, and they are animal-free. Also, because of their synthetic nature, they are ideally suited to applications in sensor devices, and they do not denature during the coupling process, as opposed to antibodies. Question for our audience today. Let's see how familiar you are with synthetic antibodies. I'll hand it over to Kelly.
Thanks, Francesco. So yeah, you should see the poll on your screen now. As you can see, we've got a few different options here. We've got aptamers, affamers, molecularly imprinted polymers, nanofittings, and potentially others as well. So I can see that most people are answering now. I'll just give you a minute to continue filling in the poll. Now, I think we've got most people answered, so let me share the results with you. And it is lovely to see that most of our attendees are indeed familiar with molecularly imprinted polymers, as well as a few others on there, which is great to see that people are actually familiar with the technology and have potentially used it as well in the past. So let me hand back over to Francesco to delve into a bit more detail.
Thank you, Kelly. So what are molecular imprinted polymers then? So a MIP, which again stands for molecular imprinted polymer, is a smart polymer that is designed around a template molecule, so a molecule that acts as a template to create an artificial binding site into the polymer. How the process of making a MIP works. Essentially, you take a template molecule, which again can be a small organic molecule, even an ion or a protein, and you essentially add monomers into the mixture in the presence of this template molecule. These monomers will self-assemble around the template molecule in such a way that the interactions between the surface functional groups of the template and the functional groups of the monomers will be optimal. Subsequently, we trigger a polymerization process. So the monomers are essentially frozen in their position, and a polymer matrix is then created. After that, we proceed with the removal of the template, which leaves behind a cavity into the polymer, which is essentially complementary to the target in terms of size, affinity, and functionalities. Sorry, size, shape, and functionalities. Now, we should think of the monomers employed here in the polymerization process as if they were the analog of amino acids. So the same way a protein is composed of amino acids, a polymer, the same way, is composed of monomers. So monomers are the building blocks of a polymer. Essentially, then the ultimate MIP generated using this process will act in a similar way to antibodies. And that's why sometimes MIPs are referred to as synthetic or artificial antibodies. Now, how nanoMIPs are produced here at MIP Discoveries. So before we delve into the more technical aspects described by the 5 steps in the bottom part of this slide, it's worth noting that the key to achieve a high affinity binder is relies on the choice of the correct monomers. And in order to do this, we make use of a very powerful molecular modeling software, which allows us to predict the best binders, the best monomers to use in the manufacturing of nanoMIPs. This will allow us to not only save time, but also increase the chances of success in generating high affinity binders. And if we think about the previous analogy that I made between monomers and amino acids, whilst there are about 20 amino acids in nature, we've got several hundreds of monomers available, which means we have a much wider choice in choosing among monomers and therefore building a high affinity binder. In the section in the center, you can see how monomers look like. Well, some of them at least, and all of them have 2 main moieties always in common. A polymerizable part, which is given by a double bond in this case, and another moiety, which is the portion of the monomer interacting with the template molecule. The kind of interactions, of course, will depend on the monomers, the solvent, and the template itself. They can range from hydrogen bonding to hydrophobic interaction, electrostatic interactions, or dipole-dipole interactions. If we now focus on how nanoMIPs are produced, so let's have a look at the steps from 1 to 5. So the template molecule, which in this case is depicted as a protein, is first immobilized onto a solid support, very often a glass bead. Why? So the reason for immobilizing the template is twofold. First of all, it will allow us not to retrieve any residual template when we collect the high affinity nanoMIPs. So it makes sure that the final product is virtually free from template. And also it orients the presentation of the molecule, which means we're going to have a much more homogeneous distribution of binding site affinity in the resulting nanoMIP. So once the template is covalently bound to the surface of the solid phase, we add the mixture of monomers. At this stage, because as we mentioned before, this is a self-assembly process, the so-called pre-polymerization complexes are formed. And again, this is driven by the energy between monomers and template. And of course, the type of functional groups present. We then trigger the polymerization. In this example, we do so with the use of UV light, which means we are making use of a photolabile initiator, but we can trigger the polymerization with other means that not involve the use of UV light. By triggering the polymerization, as mentioned in the previous slide, we essentially freeze the monomer and create a 3D entity, a nanoparticle, as you can see from those blue circles. Now, at this point, we perform a temperature-based affinity separation step, which means we need to separate between the fraction that is the fraction of particles that is free in solution and the polymers that may not have been imprinted in solution from the high affinity nanoparticles that are bound to the template on the glass piece. So we proceed with a room temperature wash, and this washes away unreacted monomers, low affinity nanoMIPs, and polymers in general. Because of the high affinity between template and the nanoparticles, the portion of high affinity nanoparticles will still remain attached to the template. At that stage, then we increase the temperature of the running solvent, and by doing so, we disrupt the stronger interactions between monomers incorporated into the nanoparticles and template itself, and therefore we can selectively collect high affinity nanoparticles. Now, this process, as my colleague Ash will explain later, is entirely scalable, and we will see later today how we've achieved scalability to commercial volumes. The typical size of these nanoMIPs are in the range of 40 to 80 nanometers, but we've improved our process, and we can also make them in a range 8 to 15 nanometers. As previously mentioned, we make use of a molecular modeling system to predict the best binders, the best monomers to use in manufacturing nanoMIPs. How this process works: so we take our template molecule, we dock it into this software, so we load it essentially into the software, and then the molecule is screened against our proprietary database of about 1,000 monomers. The software then looks at the minimum energies between the monomers and the template. And at the end, we obtain a table, a ranking of monomers, which is then used as a starting point to manufacture the nanoMIPs. Besides this information, we can also use the software to identify the optimal regions and therefore the peptides, the epitopes to use when it comes to imprinting a protein. Very often proteins are difficult to obtain or are very expensive. So one approach is not to imprint the whole protein, but choose the right epitope to imprint and then proceed with the imprinting of said epitope. We're now going to discuss a few examples of nanoMIPs used in sensor applications. In this case, on the left-hand side, you can see an example of nanoMIPs. These are our COVID-19 nanoMIPs in an LSPR sensor, so a localized surface plasmon resonance sensor. This was developed together with, in collaboration with the University of Ulster. And you can see how the nanoMIPs are able to bind the 3 variants, alpha, beta, and gamma, over other coronaviruses. In the other SPR trace graph underneath, you can see that the produced nanoMIPs are also able to bind the Omicron variant, as well as, of course, the original strain. On the right-hand side, that's another example of a sensor. In this case, is based on detecting tiny differences in temperature when the analyte binds to the immobilized nanoMIP. This was done in collaboration with Newcastle University. And you can see how the 2 graphs essentially are very comparable, and they are the dose response curves for the nanoMIPs on the left-hand side and antibodies, demonstrating the comparable performance of nanoMIPs to antibodies. Another reason why nanoMIPs are ideally suited for sensors is due to the fact that because these particles are produced in the lab, we have complete control over the surface chemistry, which means we can introduce specific functional groups for immobilization onto sensor surfaces. And in here on the left, you can see an example of electrodes modified with nanoparticles. You can see comparison between bare and functionalized electrode. On the right-hand side is an example of how our nanoMIPs are robust. So we have subject our nanoMIPs to a autoclaving cycle at 121 degrees. And you can see how the performance of these nanoMIPs prior and after autoclaving is very similar. So it's a clear demonstration of the robustness of our nanoMIPs. Another question for you, Kelly, over to you.
Excellent, thanks, Francesco. So now Francesco's given us an introduction to the technology and some of the benefits of nanoMIPs. Let's have a look at how you believe they can support your next projects. So they're potentially replacement of antibodies to improve supply chain. Could they enable shelved R&D projects to be reinitiated? Can they facilitate more robust point of care devices, enhance your sensor developments, and also support multiplex assays? So just a number of ways that MIPs could potentially support ongoing R&D projects. So I'll give you a moment for everyone to vote. And I think we've got most people have answered now. So let me share the results with you. Now, as you can see, there seems to be one that's really standing out, and that's to facilitate more robust point of care devices, which is obviously a really unique point with MIPs. They're obviously, as Francesco mentioned, they can withstand extreme conditions of temperature, pressures, various different conditions. So obviously that's a great one. It's interesting to see that everyone has picked up on that. I'll hand back to Francesco.
Thank you, Kelly. Let's now review some examples of nanoMIPs applied in sensors. This is an example of a sensor for detection of COVID-19 in the air. So it's to detect an airborne virus. This was done in collaboration with another company called Soterius. So the nanoMIPs were integrated into the sensor with the aim to detect potential contamination in public venues such as hospitals or schools. Again, it's based on the nanoMIPs that we've developed here at MIP Discovery, and Soterius has managed to achieve a sensitivity in the picogram per milliliter range, and the company is now expanding to respiratory diseases. In another example, we have been tasked to replace an antibody, a capture antibody in an assay to secure the supply chain in a top autoanalyzer. So you can see on the right-hand side how the principle works. The nanoMIPs immobilized onto the surface would then bind the target protein, and then a detection antibody would then allow the quantification of the binding event. The graph in the middle shows how comparable the performance of the antibody in red with the nanoMIPs in green is, and in yellow you've got the control given by the non-imprinted polymer. Again, this was done to replace an antibody with suboptimal stability, and as mentioned before, we've demonstrated how our nanoMIPs are far superior in terms of robustness. The MIPs and the system is currently undergoing testing and optimization at our customers. Another example of nanoMIPs we are currently working on is given by norfentanyl nanoMIPs. So they are used for the rapid detection of fentanyl in urine and saliva. As you can see from the graph on the left, we've managed to achieve a consistent particle size of about 10 nanometers. This was assessed on 3 different batches. And thanks to this size, these nanoMIPs are applicable to both sensors and lateral flow as well. They've demonstrated an affinity as measured by SPR, so first plasmon resonance, of about 8 nanomolar, with an affinity comparable to that of commercially available antibodies. On the right, you can see an early type sensor development example. So you've got a signal generated by the immobilized nanoMIPs on the sensor, which is proportional to the concentration of target analyte, versus an unrelated control molecule depicted by the red line. The nanoMIPs are currently undergoing further selectivity tests. We are also looking at improving our limit of detection, and we think we'll have this nanoMIP ready for beta testing later this year. I'm gonna now give the floor to Ash, who's gonna give you an introduction of our journal on.
Good afternoon, everyone. Thank you, Francesco, for an amazing introduction to the technology. I will be presenting the next part of this webinar, which has to do with the stepwise journey to industrialization. This is really important for us because with an amazing technology we have, this is an absolute must to support the entire IVD industry. Before I go on further, I'd just like to kind of iterate in front of you that we have a robust design control procedure in-house, which is in lines with ISO 13485. Now, as one can imagine, the very first step in this journey is, and definitely for us, is the customer needs and specifications. And during this phase, what we do is basically we look into the proof of concepts of different options or different MIPs that we could basically provide as a solution to our customer. Followed by a rigorous, and I specifically want to emphasize on it, is a rigorous lab scale optimization, which then leads to the development of protocols, not only to design or to synthesize MIP or MIP particles, nanoparticles, but also towards the analysis or towards their analysis, which then leads to the pilot scale optimization. Now, I'll delve slightly more in detail, and then you'll have a better picture to what I'm trying to say. Now, that this journey, I won't go from the very beginning because Francesco has done an amazing job for that. So I'll just start with the lab scale to the pilot scale journey. Now, in lab scale journey, the most important thing we do is not just the compositional optimization of MIP synthesis, but also the process optimization. And we do this in the very early stages, followed by the repeatability and reproducibility trial. Now, the reason being very simple, because we want to make sure that what we are making is reproducible and fits very well to the expectations of the customers. This then leads to the pilot scale process, which basically means that whatever we have achieved in the lab scale process is then replicated and scaled up at least 5 to 10 times in the pilot scale, which basically means that we repeat this cycle of reproducibility and repeatability to validate what we have actually done in the lab scale. And that has been amazingly successful. Now, I'll try and put these numbers in perspective here, where I let you basically wonder what we do in pilot scale from lab scale, and now what we are trying to do from pilot scale to industrial scale, which is a lot more than what we do in the lab scale. So we are very confident because our industrialization process is robust, and we have done that over and over again in 2 stages, one at lab scale and second at the pilot scale. So we know what our reactors are, how they behave. We know the concentration, we know the filtration, and other process control parameters. So putting numbers in perspective, if I'm talking about, let's say, sub-microgram level that we get in our lab scale, we have actually achieved not only just 5, 6, or 7 micrograms, but a couple of tens of micrograms, basically. And this is exactly what we are trying to ramp up in our industrial scale. And that's the reason why we want to scale up instead of just doing a scale up, which we have already achieved, we want to scale out by doing the parallel approach synthesis, basically. The most important thing for our success is, like Francesco did mention, is the robustness and the stability of MIPS. And this is what we are trying to achieve through our trials of stability. So we look into three aspects. One is a preservation study of MIPS. Second is the sterilization stability. And as Francesco mentioned, that we have already imposed or already challenged our MIPS in autoclave situations, and it has absolutely come victorious. We are trying to do, and we are also already putting this under sterile conditions under gamma radiations and ETO, and it's absolutely working well. We are also putting a couple of our MIPS in the accelerated and the fast accelerated stability conditions. And this is really important because this kind of confidence is needed for us in MIPS, which you already have, and this will be passed on to our customers as well. So if I have to summarize our journey to industrialization, I can say it's a well-defined and a well-planned and a robust process, which we do keeping customers' application and specification in mind, completely in lines with ISO 13485. And like I just mentioned and showed, it is absolutely and scalable process. So with this, I hand over back to Kelly for an amazing poll that you would like to participate in yet again.
Lovely, thank you, Ash. So as Francesco has given us an introduction to the technology, shared some examples of how MIPS are actively being used in IVD devices right now, and Ash has given us an introduction as to how the process is now scalable to commercial levels. It'll be interesting to see from this information, do you feel that you would potentially utilize MIPS in your future assays? So I see we've got a few people that have voted so far. I'll just give a moment for anyone else that wants to vote in the poll. And I will now share the results, and you can see from the smile on my face is very positive. We have got a lot of people that have said yes, they would be interested in looking at nanoMIP technology. So I think that's great to hear that people are seeing the potential in the technology, and this evidence of MIPS being used in commercial devices and commercial developments is really raising the profile of the technology in the industry. I see we've had a few people that have said maybe I need to know more. Obviously, we are always open to discussion, so I'll share our contact details at the end, and we can obviously get discussions rolling. So now I will hand back over to Alan, who's going to talk about the future outlook, and then we'll move into a Q&A session.
Thanks, Kelly. Future outlook for MIP Discovery. We are on this journey to industrialization. I think it's fair to say in doing things live, nerves often do get the better of us. I just want to clarify that the current lab scale is very much in the tens of milligrams, and that our aspirations are very much to be producing a gram of functional nanoMIP in a single day, which is obviously sufficiently large enough quantities to underpin a lot of the IVD companies we might want to be working with. In terms of aspirations and in terms of the MIPs that we want to produce, we want to expand into the drugs of abuse target area and leverage the ability to imprint around small molecules fully in what is a rapidly growing segment of the market. We obviously have a deep-seated desire to see FDA approval of assays enabled by nanoMIPs. It's always been a very difficult journey to move from antibodies to alternate binding molecules and get that full approval. But we believe that we're the best suited, the most capable of delivering in that area. And lastly, we want to enter the cell and gene therapy market. That's truly underpinned by the animal-free nature. In other words, endotoxin, exotoxin free, no cell line issues or contamination there, the highly scalable production processes, and that ability to sterilize nanoMIPs. and therefore ensure that they're great for closed loop systems and inline sensing or purification or even cell harvesting within the cell and gene therapy space. It really is an exciting area, and we're very keen to be a part of that. So thank you, everybody, for attending our webinar, and we're now open to answer any of the questions that have been submitted during the course of the webinar.
Lovely, thank you, Alan. We do have a few questions in the Q&A box, and if anyone else has any questions, please do add them now. The first one is, how are MIPs comparable to monoclonal antibodies?
Okay, thank you. I'll take that one. It's fair to say we're significantly closer to a monoclonal antibody than a polyclonal antibody in terms of how we recognize the target. As Francesco alluded to earlier, if we imprint around a specific peptide sequence, then our MIP will target that peptide sequence. So from that viewpoint, we do have a significant level of monoclonality, but equally, the binding pocket will have subtle variations based upon the exact positions of those monomers as they're polymerized, and therefore the strict geometric spacing of those binding events is not 100% identical to that of a monoclonal antibody. But in terms of targeting, overall affinity and performance, we're very much closer to that than we could ever be compared to polyclonal, unless, of course, a customer asks us to design something with a level of polyclonality, which of course we can achieve, but an individual nano-MIP population would be very much closer to a monoclonal in performance.
Excellent, thanks, Alan. The next question is, what surface groups are available for coupling?
Thanks, I'll let Francesco pick that one.
Thanks. Yeah, as mentioned before, because we have complete control over the surface chemistry, we can introduce essentially any functional group that the customer may want. So we've introduced carboxylic groups, primary amines, or even sulfidryl groups for attachment onto gold surfaces. So it's really dependent on the requirement and the needs of our customer.
Lovely, thanks, Francesco. The next question is, could you comment on the cross-reactivity of MIPs and how that's controlled?
I'll pick that one, thank you. So the cross-reactivity or selectivity can be controlled to a significant degree by design. It's obvious that if we target a section of a small molecule that also appears in a large molecule, then there will be some level of cross-reactivity, as there would be with an antibody doing the same thing. We have and are currently working on a number of MIPs that might be called pan family. And therefore, what we've actively done is selected the common parts of a family of molecules and targeted those for imprinting, with a view to producing a MIP that recognizes 5, 6, 7 different variants of a molecule, to the exclusion of the interference or the ones that we don't want to recognize. That part is often underpinned by the quality of our in silico modeling and how we translate that into the generation of the binding sites. If anybody listening doesn't believe that answers the question sufficiently, happy to follow up offline afterwards.
Thanks, Alan. The next question is also related to selectivity, and it's around, have we tested MIPs with real samples?
Yes, we have. We've tested MIPs in a number of sample matrices, from swab extracts, as one might imagine, with COVID, through to plasma and serum samples with some of the MIPs we've also made previously. We were happy to report that the results are as we would expect, and therefore, we're comfortable with the performance of the MIPs. We also, outside of the traditional healthcare environment, have tested MIPs in some fairly extreme conditions, namely almost completely 100% organic solvent, or oil or triphasic fluids within the oil and gas industry. So some environments that are particularly extreme compared to, let's say, human or animal sample types.
Excellent, thank you, Alan. The next question relates to the production procedure. So we've got a question here. Could magnetic beads also serve as a solid phase support? And would it be have any hazardous effects on the polymerization procedure?
Francesco?
Thank you. Yes, they can absolutely be used as solid support. There is actually a group who has which has actually exploited this method and is actually patented. And yes, they could be used instead of glass beads. The leaching of metals should not present an issue to the polymerization itself, although it might be detrimental in terms of polymerization of specific monomers, but overall, they should not present any problem.
Thanks, Francesco.
Right.
The next question is, what do you think about the future of the use of nanoMIPs in wearable sensors?
I think with wearable sensors, it's very much the case that the mode of sensing will be continuous, uncontrolled, exposed to humidity and temperature and other, let's say, non-lab condition situations. And from that viewpoint, I believe our MIPs are very well placed to work in continuous sensing. One part that Francesco alluded to earlier, in particular around the way that we produce the MIPs, is that we do the selection currently looking for high affinity. Why? Because we're trying to underpin the sort of current standard of diagnostic testing. The reality is we can actually choose to a certain degree the affinity of the nanoMIP for the template. And that's certainly a functionality that would help enable continuous or wearable sensing, whereby as the concentration increases, the binding site of the MIP is filled. And if we can transduce the signal, then we can detect that. And as the concentration drops, then the concentration will also drop in the binding pocket of the MIP and again be detectable. It's a manner very similar to, one might argue, some of the boronic acid enabled continuous glucose sensors that are now starting to hit the market.
Excellent. Next question is, could you comment on the batch-to-batch variability of production?
Yeah, like I mentioned, our robust design and the rigor with which we work, we haven't seen any major problem as far as repeatability is concerned. We have done a lot of repeatability on COVID MIPs, for example, which is one of our potential products, and it has come out completely victorious. So it's a very robust process that we put in, and the reason why we are so confident is because we try to put this repeatability and reproducibility method right from the very beginning of inception, if you like.
In context for the diagnostics industry, that means that the coefficient of variation, the CV on size of our nanoMIPs, is controlled and low. And equally, the affinity from lot to lot is, let's say, very comparable within experimental error on, say, the Biacore T200, and meets or exceeds the consistency that we could get with a monoclonal antibody under similar testing circumstances.
Excellent, thank you, Alan. The next question is, how do you solve the problem of surface physical absorbance? Technical one for you.
Does that mean we're talking, I'm going to talk hypothetically here, of something from the sample adsorbing onto our MIPs? Or are we talking about our MIPs absorbing to a surface? And I think I'll pick both of those. So putting MIPs onto surface or adsorbing does not happen in the same way that it would with a protein or an antibody. Our MIPs don't denature, they don't try and take that lowest energy state on a solid surface in the same way that a protein would. And therefore, we do typically use some controlled chemistry and take advantage of the chemical groups we put in to attach MIPs to surfaces. If we've got the MIP on a surface and we're working in let's say a very complicated or highly proteinaceous environment, then again, we can take advantage of the chemistry. I know that Francesco and the team here have grafted non-specific binding reduction molecules onto surfaces of our MIPs and incorporated them into the synthesis process. That's certainly not something that an antibody would tolerate easily and still remain suitably functional. So we can use the chemistry knowledge we have to address those particular situations.
Excellent. And we've got quite a few more questions, so we'll see if we can get through them all. The next one is, if you have a target with no functional groups, how could you prepare MIPs for it?
Jessica.
Well, if there is absolutely no functional group, so we're talking about alkane or alkyne chains, then technically the chances of getting a high affinity nanoMIPs are quite slim, because as mentioned in one of the previous slides, you need to have functional groups interacting, between the monomers and the template itself. If the template is simply made by carbon and hydrogens, there is very little that you can exploit there. The resulting MIP would have most likely low affinity, which, depending on the application, that can be something that would still be usable. If we're talking about detecting alkanes or hydrocarbons at very low concentrations in an environment with other alkanes or hydrocarbons, so with high affinity and selectivity, then that's not something that is easily doable.
Excellent, thank you. The next question is around in vivo measurements. So have you done any in vivo measurements regarding the nanoMIPs, and have you seen any matrix effects?
I'll hand to Francesco.
Well, here at MIP Discovery directly, no. With help of some of our collaborators, yes, we've explored a few aspects. First of all, how the nanoMIPs are distributed within a mouse, and how they are cleared from the body. And then in a different work, we've explored how we can target, or whether we can target in vivo certain cell populations. In this case, it was a work on senescent tissues. And yes, we've got a publication on targeting senescent cells. And again, this was done in mice. So we're happy to send over the publication in case whilst the question is interested.
Excellent, thank you. The next question is, can nanoMIPs be used in breath analyzers, and are the problems of humid interference resolved in this case?
So humidity, in terms of having a binding interaction take place, usually a small amount of water is beneficial, particularly around the target. If we're talking breath sensing in the strictest gas sense, then obviously it will be somewhat challenging. But if we're talking about the sorts of molecules that are in the exhaled breath condensate, of which there's a significant interest at this point in time, then our MIPs would ideally work in a sensor there and have a significant lifetime in use. Some of how that might be enabled might come down to the interest from a potential customer, their underlying sensing platform, and the ability to get that breath sample to the sensor interface. But I think, as with any other sample matrix, it should be entirely feasible. And of course, the COVID test with Sotirius in terms of that environmental sensor is not massively dissimilar. Now, that's virus particles that might be in exhaled breath droplets landing on a sensor surface and being detectable. It's just not straight in the direct breath stream per se.
Excellent. And then the next question is, in the synthesis process, have you synthesized MIPs on nanoparticles? And the next question is very similar. Do you first synthesize the MIPs and then bind them to nanoparticles, or do you synthesize the MIPs directly on the surface?
Francesco.
Yeah, I mean, the approach is absolutely versatile. We can do both. We can either make a preformed nanoMIP and then exploit the functional groups on both the nanoMIP and the counterpart, let's say a gold nanoparticle or polystyrene colored particle, and then covalently link them together. This is one option. But there is also the option of grafting a MIP layer around a seed, and this seed can be, again, a gold nanoparticle, a magnetic particle, the particles that you may be interested in. Clearly, the chemistry behind will be slightly different, but the imprinting process will be essentially the same.
Excellent. Thank you, Francesco. So we are running out of time. I think we have captured every question, but if there is any we have missed it in the questions there, please do get in touch. Our contact details are on the screen now. So if you would like to discuss MIPs in any more detail, please email us at inquiries@mipdiscovery.com and a member of our team will obviously get back to you. I'd like to say a big thank you to our speakers today. They've given a very insightful presentation, and I hope that it has got you thinking about how MIPs could be used in your next generation devices as you develop them. Thank you very much for all our attendees, and as I say, please do get in touch if you'd like to discuss any aspects of the presentation in more detail. Thanks very much for joining us, and hopefully we'll see you on our next webinars. Goodbye.